Thanks for the invitation. It's a fantastic workshop. Yes, I was going to talk about this theory. This is, this is joint work with a graduate student in my lab, Peiran Gao, and Eric Troutman, a graduate student in Krishna Shenoy's lab, who collected the data. These are more acknowledgments about people in my lab and our funding sources. Actually, I put this new slide in my talk just during the last talk, actually, the very interesting last talk on the theory of deep learning. We've actually also been working on the theory of deep learning, and I could have talked about that today, maybe I should have. But I just wanted to outline some references here and just give you some take-home messages. So we've also been thinking about why deep learning works, and we have some results on exact solutions to the, some results on exact solutions to the dynamics of learning in deep networks that motivate certain initializations. We've also been thinking about saddle points in energy landscapes, and that actually motivated a new type of algorithm to rapidly escape saddle points. We've actually been using ideas from non-equilibrium thermodynamics to train very, very deep generative models by essentially reversing the flow of time in a diffusive random walk that destroys structure and data. So by reversing that destruction of structure, we can start from noise and generate data. We've also just very recently, we've come up with a theory of the expressivity of deep neural networks that connects it to the theory of chaos. We can show that deep random neural networks create very highly curved functions whose curvature can grow exponentially with depth, but not width. And we've also applied deep neural networks to educational data and also retinal data, which I haven't put up here. Anyways, you can find all of these papers on our website, except the last couple, but they're up on the archive. I just haven't updated the website yet. In any case, with that interlude aside, let's talk about what I was actually going to talk about. So what I wanted to discuss was a major conceptual elephant residing within almost all work in systems neuroscience, and that conceptual elephant is the following. How can we record about 100 neurons in regions deep within the brain, far from the sensory motor periphery, and obtain scientifically interpretable results that actually relate neural activity to behavior and cognition? The fact that we can do this is remarkable, because these brain regions have about 10 to the 6th to 10 to the 9th neurons, about 5 orders of magnitude more than we can record. So a natural question is, how has systems neuroscience been as successful as it has been in this vastly undersampled measurement regime? Or is the situation much worse than we think? Are we completely misleading ourselves? Can we trust any of the scientific insights we get from the brain by recording so few neurons? Okay. So lest you think that there's an experimental solution to this problem, while it is the case that neuroscience has been experiencing its type of Moore's law growth or exponential growth in the number of neurons that we record, the doubling time has been about 7 years. So if we extrapolate current trends, it will require about 122 years to get the requisite 5 orders of magnitude more neurons to completely record all the neurons in a relevant brain circuit. So what we need now, I can't wait 122 years. Maybe some of the undergrads or grad students could. But we need a theory of neural data analysis that tells us how and when statistical analyses applied to a small subset of neurons reflect the collective dynamics of the much larger unobserved circuit they are embedded in. Okay, and this is, of course, a question that extends far beyond the writ of neuroscience. It's really a question of what can you say about dynamical systems by observing a small subset of their degrees of freedom. Okay. So what's the type of data that we're looking at? So we're looking, we're going to apply our theories to data recorded from monkeys while they're reaching. And so this is data recorded from monkeys reaching to several different directions across 2 different reach extents and 2 different speeds. And what you do is you record isolated single units from the monkey, and you average their firing rate, spikes per second, over trials, and you get these smooth trial average firing rates. And they're quite heterogeneous. This is 1 neuron, this is another neuron, this is another neuron, and there's about 100 other neurons like this. Okay? It's a really heterogeneous, complicated data set on its surface, and so the question is, how are such data sets analyzed? Of course, dimensionality reduction is a common way of analyzing them. For this audience, I don't need to introduce principal components analysis, but basically you can do principal components analysis on the neural trajectories and decompose the pattern of neural activity, all neural activity patterns occurring through the experiment in terms of a time-dependent linear combination of a static set of basis patterns. And you can look at the trajectories that you get in pattern space or principal component space. And this often yields a very insightful dynamical portrait of the computation that the circuit is doing. Here are some examples of principal component portraits or other types of dimensionality reduction procedures describing what the circuit is doing. So for example, in the locust antennal lobe, this tells us that neurons in the locust antennal lobe circuit contains a lot of information about odor in the transient dynamics long before it approaches a fixed point. Here, in a monkey that's supposedly doing a tactile discrimination task, this tells you that most of the variance in the monkey's neural data goes towards accounting for the passage of time as opposed to the different frequencies it's supposed to discriminate. Here, in monkey prefrontal cortex, in a recent paper, they discovered a novel method for doing context-dependent decision-making in a monkey by looking at the geometry of these portraits. And here, you can actually record from the entire brain of the zebrafish and fit the data into 3 dimensions, which is another kind of a mystery. Okay? So the fundamental conceptual questions we'd like to ask are the following. Can we trust these dynamical portraits of circuit computation, given we're recording so few neurons? For example, how would the shape of these portraits change if we recorded more neurons? Oftentimes, their dimensionality is low, much lower than the number of recorded neurons. So the question is, what's the origin of this simplicity? Would their dimensionality increase if we recorded more neurons? And again, what, if anything, can we learn about large dynamical networks at such overwhelming levels of undersampling? We'd like to obtain a predictive theory of experimental design that can tell us how many more neurons we should record as we go forward to do more complicated experiments. And how should this number depend on the properties of neural activity and the task that the animal is asked to do? So let me just give you the take-home messages, and then we'll work through why these take-home messages are true. So our theory is predicated upon sort of a definition, a mathematical definition of neural task complexity. It takes into account 2 features of experimental design. 1, the behavioral complexity of the task that the animal is asked to do, and 2, the smoothness of neural activity patterns. And with this mathematical definition in hand, we can prove that the dimensionality of neural data is upper bounded by this neural task complexity quantity, right? So then we can go back to motor cortical data and measure the neural task complexity. And what we find is that motor cortical data, when you record about 100 neurons, the dimensionality of the data is about 10, a little bit less than 10. But it turns out that that's also what the neural task complexity is. So essentially, motor cortical data is as high dimensional as possible given neural task complexity. And this suggests that in various brain initiatives where the push is to record more and more neurons, this suggests if you simply record more neurons without fundamentally changing the task, you won't obtain richer data sets, at least as measured by the dimensionality of the data set. You'd have to go to more and more complex behaviors, really, to leverage the power obtained by large-scale recordings. On the measurement side, we can give conditions for the accurate recovery of these dynamic portraits, and what we do that by making a connection between the active electrophysiology, recording a random subset of neurons, and the theory of random projections, which of course many of you are familiar with in machine learning and mathematics. And this tells us, fortunately, that our past results, we can actually trust them. They're likely to be accurate despite recording so few neurons, which is good news. Okay, so what is this idea of neural task complexity, and why might it plausibly upper bound complexity? It's actually a very simple measure. Just let's illustrate it through a couple of examples. Let's imagine that a monkey is reaching to a certain point. Okay? The reach takes a finite amount of time, call it capital T. For this particular experiment, that time is 600 milliseconds. And so you can record from a whole bunch of neurons, and these are their superimposed trial average firing rates. So this is a trajectory in a high-dimensional neural space. But the number of linear dimensions it explores, which is what PCA measures, can't be that high because this trajectory doesn't change infinitely fast. It has a certain autocorrelation time tau. So you have to wait an amount of time tau before you explore another linear dimension, another time tau, and so on. So the number of dimensions should be approximately capital T over tau. Okay? What if a monkey is doing a slightly more complicated task, for example, reaches to multiple angles? Then the task manifold is parameterized by a cylinder, and the trial average neural data is fundamentally a smooth embedding of the cylinder into neural activity space. Now, the argument that I gave for time at a fixed angle also holds for angle at a fixed time, yielding a dimensionality of about 2 pi over delta, where delta is the spatial autocorrelation length of neural activity patterns across angle. And if we imagine just roughly and intuitively that the explorations of this manifold across time and angle are uncorrelated in a way that can be made precise, then we expect that the total dimensionality should be the product of the 2 dimensionalities, or at least an upper bound on the dimensionality should be related to the product. So we can make these very simple intuitions precise in the following theorem. So imagine you have a bunch of task parameters that vary over some ranges L1 through Lk, with neural autocorrelation lengths lambda 1 through lambda k. Then this neural task complexity is essentially the volume of the task manifold divided by the volume of a unit correlation cell across the task manifold. And with some constant, our theory provides a quantitative way to extract these neural correlation lengths and the proportionality constant C, such that we can prove that the participation ratio of the PCA eigenvalue spectrum, which is a measure that varies from 1 to n, this is 1 if one eigenvalue is active and all the other eigenvalues are 0, and it's n if all the eigenvalues take the same value. It roughly corresponds to 70% of variance explained in a wide variety of spectra. We can show that this measure of neural dimensionality is upper bounded by the minimum of neural task complexity of the number of recorded neurons. We don't have time to go into the proof, but the essential proof idea is we systematically destroy structure and data, and under each destruction of structure, we show that the dimensionality increases, so that when the data approximately behaves like a completely random data set subject to autocorrelation lengths, then the dimensionality is this quantity, right? So because this is the dimensionality of a data set whose structure has been destroyed, this dimensionality this number is an upper bound on the dimensionality of the original data set. Okay, so sort of smooth random walks, for example, saturate this upper bound. Smooth random manifolds saturate this upper bound. Okay, so now what implications does this have for the interpretation and design of neural experiments? So again, this is an experiment where a monkey is reaching to a target, and we analyze the neural data during the dynamic period of activity while the monkey's reaching. Okay? We look at multiple single unit recordings from the monkey's brain, and again, there's 109 single units. There's another a slightly more complicated data set where we get similar answers, so I won't talk about that. Just to throw out some numbers, in primate motor cortex, there are about 100 million neurons controlling 650 skeletal muscles. In these experiments, 100 neurons were recorded. The PCA dimensionality across all 8 reaches is 7, so it's much less than 109. And then the PCA dimensionality just for one reach is 3.3. Okay? So where do these numbers come from? Can we come up with an appealing, intuitive, and quantitative understanding of where these numbers come from? So what we can do is we can compute this neural task complexity, and we find that it's 4.2 and 10 respectively. So these numbers are almost as large as these numbers, right? So the implication is that we shouldn't think about neural dimensionality as small, but almost as large as possible given the simplicity of the task, so a simple reaching task that the monkey is asked to do, right? So, the business of doing theoretical neuroscience is making predictions that can be tested in data. So this theory does make some predictions. It suggests that if you vary the task complexity by varying the duration of time over which you analyze the data, right, then the dimensionality should vary linearly with time. Okay? Prediction number 2, if you drop a large number of neurons in the data set, the dimensionality should essentially remain unchanged, because the dimensionality is essentially constrained and upper bounded by the complexity of the task, not the number of neurons. Okay, so these predictions bear out. So here you do see that the dimensionality grows with time linearly, and it roughly hugs the upper bound, the theoretical upper bound. And also, if you drop 60% of the neurons, the dimensionality essentially remains unchanged. Okay? So that's sort of a better understanding of neural dimensionality. Okay, but now what about the shape? What about the shape of these portraits? And for this audience, I think this will be very intuitive. The question we'd like to ask is, when can we get the right answer despite recording so few neurons, i.e. under which conditions would the shape of a neural manifold remain the same if you only recorded a subset of neurons? So if the neural manifold is randomly oriented with respect to the single neuron axes, in a way that again can be made precise, but I won't do it here, then it's sort of intuitively clear that it doesn't matter the simplest situation is a system where you have 3 neurons, and for whatever reason, neural activity patterns are constrained to be on a one-dimensional manifold, and you only get to record a subset of 2 neurons. So here it's intuitively clear that it doesn't matter which subset you record, you get the right answer, in the sense that the geometry of this manifold is independent of which subset of neurons you record, and it's similar to the geometry of the manifold you get if you recorded all the neurons in the circuit. Of course, it doesn't have to be that way. You could have a situation where one of the neurons is doing all the work, so then any subset you record better include that neuron. Okay? So how do we make this theory quantitative enough that we can come up with a quantitative prediction that we can test in experiments? So what this suggests is that if the neural manifold is indeed randomly oriented with respect to single neuron axes, an experiment that we routinely do becomes equivalent to an experiment that we cannot yet do. And that's basically an experiment where we record from a random subset of neurons becomes equivalent to where we record, okay, so here we record from M neurons, becomes equivalent to an M-dimensional random projection of neural activity. So if you had a magical electrode that recorded from one random linear combination or another random linear combination, then these two experiments would become equivalent. Why? Because the first experiment corresponds to projecting the neural manifold onto a coordinate subspace, whereas this corresponds to projecting the neural manifold onto a random subspace. And if the neural manifold is randomly oriented to begin with, then obviously the two experiments are equivalent. So this suggests a random projection theory of neural measurement. Okay, so what does random projection theory tell us? So clearly, often what happens is we get signals that live in a very high-dimensional ambient space, but they often concentrate on interesting low-dimensional linear manifolds. And a natural question would be, if you projected these manifolds down to a random subspace, when would you preserve the pairwise geometry of these points? So a measure of the preservation of pairwise geometry is the distance in the low-dimensional subspace minus the distance in the high-dimensional space, divided by distance in the high-dimensional space. So this is the fractional error in pairwise geometry. And a natural measure of the quality of the projection is the worst case distortion over all pairs of random points. So, there's various ways of analyzing particular manifolds. This, for example, the union of sparse planes leads to compressed sensing. Discrete points leads to the J lemma. But what we're interested in is smooth manifold theory. So there's an old result by Baraniuk and Waking that says that as long as the number of random projections is 1 over epsilon squared times the intrinsic dimensionality of the manifold, k, not the ambient dimensionality, times the logarithm of some quantity related to the curvature of the manifold in the volume, then the worst case distortion will indeed be order epsilon with high probability over the random choice of projection A. Okay, very recently, we haven't submitted this yet, we've actually come up with a much simpler formula for this theorem that actually leads to, there are some constants in this work that are very large, and we can get rid of those constants now by looking at random projections of random manifolds. That should appear in the archive very soon. So we're actually using a slightly newer theory, but the basic qualitative structure of the prediction is the same. So if we think about this quantity, curvature times volume, right? If we remember neural task complexity, which is volume divided by autocorrelation length, these are quite similar in the sense that curvature is like 1 over correlation length roughly. So we can replace this quantity with neural task complexity, essentially, the same quantity that upper bounded dimensionality. Okay? So then this makes a very precise and striking prediction that we can test in neural data, which is the following. Let's say, I would like to do the most complicated experiment possible and record all the neurons in the brain, but we don't have that. We have this reaching task where we have 109 neurons. So what I could do is I could, for a particular level of task complexity, which corresponds to how much time I keep in the analysis, I could compute the PCA portraits or the dynamics using all the neurons, or I could reduce to a subset of neurons, right? And I could measure the distortion from at this point relative to this point, or say at this point relative to this point. What the random projection theory says is that the number of neurons needed to achieve a certain distortion is proportional to the logarithm of the neural task complexity. So what this says is isocontours of constant distortion are straight lines in a plane formed by the complexity of the task, but the logarithm of the complexity of the task, and an axis which is linear in the number of neurons you keep in your analysis. If you allow yourself to be more generous, you get a different isocontour, where for any given task complexity you record more neurons, but then the isocontour should be a straight line here. So basically, to keep the same level of distortion, the number of neurons need only scale logarithmically with the task complexity. This is fantastic news. Now, we can test this now in real neural data. Now, it's important to keep in mind that this test isn't an empty mathematical test of a theory that's already been proven. It's a test that the neural data conforms to the assumptions that that theory requires. And the assumption is that the projection is indeed random. Within the neural context, this means that the manifold of neural activity patterns explored by the monkey while it's reaching is randomly oriented enough with respect to the single neuron axes that random projection theory constitutes a good theoretical model for the act of doing systems electrophysiology. Okay, so remarkably, this theorem works out. Okay, so we did exactly what I said we were going to do. We varied the neural task complexity by varying the amount of time in our analysis, and this is a logarithmic axis. And we varied the number of neurons in our analysis, and we computed the distortion relative to what we would have gotten had we kept all the neurons in our analysis for that amount of time. And we find that the isocontours of constant distortion are indeed straight lines in this log linear plot. Okay? So this does verify this does lend credence to the idea that the monkey organizes its neural activity patterns in such a way that they can be easily read out by looking at a subset of neurons. Okay? That's fortunate for us. One might ask a teleological question of why did the monkey's brain do that? I think it does that because downstream neurons need to solve the same problem. Downstream neurons only get to send a small number of synapses up to upstream regions. And so whatever downstream neuron, whatever information it needs to get out of the brain region, it has to do with a small number of measurements. So because the brain organizes dynamics to be easy for downstream processing to occur, a fortunate corollary of that was it made itself easy for neuroscientists to understand the brain itself by recording a small subset of neurons. But that's, of course, speculation. So let me give you, although I think it's reasonable speculation. Anyways, so let me just give you the take-home messages. So when we measure neural dimensionality, we should really compare it to this upper bound set by the complexity of the task. And if it's equal to that, then your dimensionality is constrained by the complexity and smoothness of your neural recordings, complexity of the task and smoothness of neural recordings. If it's less than, and only if it's less than, have you then discovered an intrinsic dynamical constraint in the circuit that goes above and beyond the task and smoothness constraints. And then this tells you that subsampling can indeed recover accurate dynamic portraits where neural activity patterns are highly distributed. We've done actually a lot of work beyond this, going beyond the single trials. So this was for trial average data. If you have single trials, then there's lots of interesting new phenomena, right? For example, how well can we decode behavior in single trials? Essentially, can we come up with a theory of why in brain-machine interfaces you can use a simple linear decoder and to a very good approximation decode the person's intentions or actions and use it to guide a prosthetic arm. An interesting thing that occurs both in monkeys and humans is that when you ask a monkey or human to do something, they're often thinking about other things, and those things vary from trial to trial. So there can be latent cognitive subspaces of activity that vary from trial to trial. And they're not just noise, they're actually the brain thinking. All right? We'd like to discover those. For example, your attention can wax and wane during a talk. And that waxing and waning, that's probably an example close to home. But that waxing and waning can actually be deduced by doing latent variable dynamics on, say, neural recordings from monkeys. So we can learn the latent structure of unobserved latent cognitive variables. And we want such a theory to help us with experiments before they're done. This leads to all sorts of interesting analyses related to, and of course I won't have time to explain any of this, I wasn't planning on it, but it relates to all sorts of interesting theory and analyses relating to free probability, random matrix theory, and so on and so forth. But it actually suggests a really interesting phase transition that yields a trade-off between different resources. Let's say we'd like to recover latent cognitive subspaces, and we have different amounts of resources, the number of neurons we can record, and the number of trials or samples we use to collect data to estimate the subspace. There's a hyperbolic phase transition boundary such that when you're on the right side of the boundary, you can correctly recover the dimensionality and accurately the subspace overlap. So this is in simulations where we know what the correct subspace is. And there's this hyperbolic phase transition boundary, which is very intriguing from a neuroscientific perspective because this suggests a trade-off between two vastly different resources, right? Going up here involves creating new technology that records more neurons. Going along this direction requires creating new technology that allows you to record stably for longer periods of time, or design tasks that allow the monkey to work for longer periods of time. So these are two vastly different resources, but there's a nice trade-off between them. Does this trade-off actually exist in neural data? And we tested it And we find a similar hyperbolic phase transition boundary in real neural data. And I mean, I can't really go into the details here. So there's a whole bunch of other stuff on, this is really an hour-long talk. We can ask also a very interesting question on, a really interesting question of what if you have a recurrent dynamical system and you only get to record a subset of its nodes? That dynamical system, if it has low-dimensional state-space dynamics, when can you recover the subspace correctly? And again, there's a hyperbolic phase transition boundary in the number of neurons that you record and the amount of time that you record. And it also works for nonlinear networks. But anyway, let me just end. So the lion's share of the work was done by my grad student, Peiran Gao. There's a big sucking sound from academia to industry. He had an offer at Harvard to do theoretical neuroscience with some of the best people in the field, but instead his new boss is now Elon Musk. He wants to go to space, so he's working at SpaceX. And Eric Troutman helped out in Krishna's lab. This is the funding. You can find a lot of our papers on this website, and this is Twitter. Thanks.